Good morning to all. Today we are going to discuss about regulation of gene expression in eukaryotes. In this module, the two topics contained with antisense RNA strategies for regulating gene expression. Second one is siRNA and miRNA in regulation. The first topic is antisense RNA strategies for regulating gene expression. Antisense RNA molecule represent a unique type of DNA transcript that comprises 19 to 23 nucleotide and is complementary to mRNA. Here, what is meant by antisense RNA? A antisense RNA means that represent type of a DNA transcript that comprises with 19 to 23 nucleotide and it is also complementary to mRNA. Antisense RNA play a crucial role in regulating gene expression at multiple levels such as at replication, transcription and also translation. Here antisense RNA that it can play regulating gene expression at a different multiple levels such as in the case of replication, transcription and the translation. In addition, artificial antisense RNA can effectively regulate expression of related genes in host cells. That it can play, that means artificial antisense RNA that it can regulate expression of the genes in the case of host cells. Antisense RNA are single-stranded RNA molecules. It is a single-stranded RNA molecule that it can bind and inhibit a specific mRNA translation to protein. Okay. Then naturally occurring antisense RNA has been identified in E. coli which carry an R1 plasmid and control Hock or Salk system that involves the production of toxic and antitoxic production. That means naturally occurring antisense RNA that identified in firstly E. coli. In E. coli that carried R1 plasmid and it can also control the Hock and Salk system. In that it involves with the production of toxic and antitoxic product. Next topic, that means in antisense RNA it can also affect and applications. Then we can study about effect and applications of antisense RNA in antivirus treatment. The first one is the virus that including DNA and RNA viruses. Mostly as we all know, which all are they? HIV, HCV, like that. Here viruses that including DNA and RNA viruses are most successful forms in the earth. In that case, for example, HIV, that is human immunodeficiency virus, then HCV, that is hepatitis C virus, then influenza virus, that is IV, etc. In second effective treatment is anti-cancer treatment. Cancer is not only one of the biggest global killers in humans but also one of the fastest growing fatal diseases. As we all know, cancer is the fastest growing diseases, and here the antisense RNA that can help in the case of cancer diagnosis and treatment. Okay, then third one is the in plants used in inhibition of fruit maturation, virus resistance, flower coloration, starch synthesis, male fertility and sterility, and also suppression or elimination of expression of genes involved in the synthesis of harmful substances in food. Here the effect and application, the first one is in the case of viruses that including both DNA and RNA viruses. Then second one is the anti-cancer treatment for helpful for predict cancer and also cancer diagnosis and in the case of treatment. And third one is in the case of plants that is also used in the case of virus resistance, flower coloration, starch synthesis, like that. For example, these all things. Then it is can also suppress or eliminate in the expression of genes involved in the synthesis of harmful substances in the food. Next topic is siRNA and miRNA in regulation. First one is the siRNA regulation. siRNA means, as we all know, what is meant by siRNA. siRNA is the small interfering RNA. It is able to regulate the expression of genes by a phenomenon that is known as RNAi, that is RNA interference. what is meant by siRNA, that is called as it is a small interfering RNA, and it is able, that is ready to regulate the expression of genes by the phenomenon or process is known as RNA interference, is called as RNAi. The discovery of RNA interference, first in case of plants and later in mammalian cells. The first identification or discovery of the RNA interference in the plants, and the second one later is the mammalian cells. siRNA has gained attention as a potential therapeutic reagent due to its ability to inhibit a specific genes in many genetic diseases. siRNA can be used as tools to study a single gene function both in case of in vivo and in vitro, and there is an attractive new classes of therapeutics, especially against undruggable targets for the treatment of cancer and other diseases. RNA interference, the biological mechanism by which double-stranded RNA induces gene silencing by targeting a complementary mRNA for degradation. It is a tremendous innovation in universal therapeutic treatment of disease and also revolutioning the way researchers study gene function. Next is the mechanism of RNA interference. RNA interference involves the processing and cleavage of longer double-stranded RNA into siRNAs, generally bearing two nucleotide overhang on 3' end of each strand. Okay, there is enzyme, that is enzyme responsible for this process. That enzyme is called as RNase 3 like enzyme termed as dicer. Okay, when formed, siRNA are bound by multi-protein component complex referred to as RISC. RISC means it is a RNA induced silencing complex. Within RISC, RNA-induced silencing complex is siRNA strands are separated, and strands with more stable 5' end or 5' is typically integrated to active RNA-induced silencing complex. Antisense single-stranded siRNA component then guides and aligns siRNA complex. Okay, antisense single-stranded siRNA component guide and aligns that RNA-induced silencing complex on target mRNA and through the action of catalytic RISC, that is the what is meant by RISC, that is the RNA induced silencing complexes, that protein and member of a arbutin family in mRNA then cleaved. Okay, finally that mRNA will be cleaved. Next is the miRNA regulation. miRNA means it is a microRNA or small non-coding RNAs that function as what is the function of miRNA, that is guide the molecules in RNA silencing. Okay, what is meant by miRNA? miRNA is a type of microRNAs, that means it's a small non-coding RNAs, and the function is guide molecules in RNA silencing. Biogenesis of miRNA is under tight temporal and spatial control. Dysregulation of miRNA, what is meant by dysregulation? That is the impairment. Okay, then impairment of miRNA is associated with many human diseases, particularly cancer and neurodevelopmental disorders. Regulation takes place at multiple levels including transcription, then Drosha process. That what is meant by Drosha process? That is a catalytic subunit of microprocess complex and that initiate that microRNA. Then the dicer process and RNA editing, then RNA methylation, uridylation, adenylation, then argonate modification and RNA decay. Here what is meant by argonate modification? Argonate means it is a protein family member and it can play a role in regulation of gene expression. Most miRNA are transcribed from DNA sequences into primary miRNA and processed into precursor miRNAs. This miRNA are critical for normal animal development and are involved in a variety of biological processes. Here, extracellular miRNAs reported as potential biomarkers for a variety of diseases and serves as signaling molecules to mediate cell and cell communication. For what? This cell and cell communication, the serves as a signaling molecules or serves as a signaling molecules to mediate for what? The cell and cell the communication. Then next one is the regulation of miRNA. Here we studied miRNA regulation and how they regulate miRNA. These microRNAs are the small molecules of 18 to 23. Here the siRNA is the which type here? Not siRNA. It is an antisense RNA. Antisense are that represent the 19 to 23 nucleotide. Here that miRNA, that is the 18 to 22. Okay, 22 nucleotide, and that is serves as a important regulators of gene expression at a post transcriptional level. And one of the mechanisms through which miRNA regulate gene expression involves in the interaction of their seed sequences, primarily with 3' and most rarely with the 5' end of mRNA transcribed from the target genes. Then next one is profiling miRNA expression can be an important tool for diagnostics and treatment of diseases, helps to analyze and systematize that current data, explains reasons for changes in expressions of miRNAs. It covers both in the case of transcriptional and post-transcriptional mechanisms of regulation of miRNA expression, as well as the effect of endogenous and exogenous compounds on miRNA expression. Okay, here we are concluding. Before that, what is here today? What we will study. First one is the antisense RNA strategies and their effect and applications. In the case of antisense RNA molecule, that is an DNA, that is a type of DNA transcript comprises with 19 to 23 nucleotide, and it is also complementary to mRNA. Then some effect and application in the case of viruses, plants, and in the anti-cancer treatment. Then next one is the siRNA, and what is meant by the miRNA regulation. siRNA means it is a small interfering RNAs, and it is regulate the gene expression in the phenomenon. It's called as RNA interference. Then next one is the miRNA. miRNA is the micro RNAs, and it's a small non-coding RNAs, and main function is to guide the molecules in the RNA silencing. Then next one is the reference is the genes 9 Benjamin 11. Okay, then today we are concluding. Thank you.